in the Hess Lab, we're currently focusing on a lot right now is rumen fermentation and how to mitigate methane. So what we're doing a lot is we're trying different additives and then we're putting them into our in vitro systems to test them out, see if any of them can either inhibit methanogens or if any of them can alter the microbiome and produce less methane. The reason why we're doing in vitro first is because we want to make sure that what we're adding to the feed is safe for the animals first. So we can see what we're going to expect if we were to do an animal live trial. You start with just the SBR first, and then we'll do the treatment after. Okay. And then I'll go with you again. We're testing different agricultural byproducts that are produced, and a lot of that goes to waste, either into compost and landfills. And a lot of these times, they are generating a lot of greenhouse gases, plus the greenhouse gas being emitted from the ruminants. So what we're trying to do is basically kill two birds with one stone. So reducing compost and landfill waste, and also using them as a feed that can potentially have bioactive compounds to reduce methane in the ruminants as well. We finished setting up all of our Ankom systems, and then we put them into the water bath, hooked up to our gas bags. Turn on our water bath, and the water bath is also set at 39 degrees Celsius, which is the temperature of the rumen. It's mimicking the contraction of the rumen, so it's helping all these microbes inside the vessels move around, attached to the feed, and then when it hits a certain pressure, it will actually open up the valve, and then gas will travel up to the gas bags, and we'll collect it and then use our gas chromatography to measure how much methane and carbon dioxide was produced over 24 hours. And it's really hard to measure gas measurement in actual animals because they're always moving around and you have to basically keep it in one spot. But when we put them in these gas bags, we can basically have a more controlled environment. We mimic the cow in multiple ways. One is we collect rumen fluid from the animals, so that's basically the rumen. Then we also create the artificial saliva buffer, and we also have our feed, which is basically what we're feeding to the microbes. Here we're just processing the rumen fluid that we've collected. So yeah, in the rumen fluid, there's a lot of bacteria that can break down the fibrous part of the diet. There's other microbes in there as well, such as archaeas, which are the ones that create methane and methanogens. We also have some protozoas in there, and they all work together in this fermentation process. We work with a lot of different byproducts that are grown in California specifically. So for example, we've worked with the wine industry because when they press the grapes, a lot of grape pomace are left over. And in the end, the goal is to basically compare all these different byproducts and how it alters fermentation and what microbes are present. And another thing we want to compare is the chemical composition of these different byproducts and see which one is more nutritious for the animal as well. Because it's not just ruminants that have microbes, right? Humans also have microbes in your intestines. A lot of these microbes, they can relate back to human health. So I think it's an interesting field when we go from microbes in the rumen and how they affect animals to go into like human microbes and see how they affect humans.